All right, today we are diving deep into the world of enzymes. These are the tiny microscopic machines that honestly make all of life possible. See, without them, the chemical reactions that keep you alive and breathing would just be way, way too slow. So, what are they, and how on earth do they work?
So let's start with a really big question. Have you ever just stopped and wondered, how does life happen so fast? I mean, the reactions you need to digest your food or even heal a paper cut. On their own, chemically speaking, they're incredibly slow. So what's the secret? What's speeding everything up?
So, here's our game plan for this explainer. First, we're going to nail down what biological catalysts actually are. Then, we'll get into the famous lock and key model. It's the best way to picture how they work. After that, we'll look at two really critical factors that mess with them: temperature and pH. And finally, we'll wrap it all up with a quick recap of the main rules. Let's get right into it.
Okay, so what exactly are these things we call biological catalysts? To really get it, we need to get a couple of key definitions straight in our heads first. First off, in the world of chemistry, a catalyst is basically anything that speeds up a reaction without actually getting used up itself. You can think of it like a really helpful tool on a factory assembly line. It helps you build the product way faster, but the tool itself is totally unchanged at the end, ready to go for the next job. So how does that relate to us, to biology? Well, an enzyme is simply our body's own personal catalyst. These incredible things are made of protein, and their whole job is to speed up all those metabolic reactions that keep us alive. Without them, honestly, nothing would happen fast enough to sustain us. And get this, they are total specialists. You'll notice that their names almost always end in ase. For instance, amylase, it only tackles starch. Protease, it only works on protein. And lipase, you guessed it, it breaks down lipids or fats. And then you have these powerhouses like catalase, which takes dangerous hydrogen peroxide and breaks it into harmless water and oxygen at a mind-boggling speed. Okay, so we know they speed things up, but how do they actually do it? Well, to understand the mechanism, we can use a really simple but brilliant analogy. It's called the lock and key model. I want you to picture the enzyme as a lock that has a very, very specific shape. The molecule it needs to work on, we call that the substrate, is the key. And for anything to happen, the key has to have the absolute perfect shape to fit into that lock. No other key will do. The whole process is like a simple three-step dance. First, the substrate, our key, finds the enzyme's special groove. Second, it slots in perfectly, and for a brief moment, they bind together, forming what we call an enzyme-substrate complex. Then boom, the reaction happens. The substrate is changed into new things, the products, and they get released. This frees up the enzyme to do the exact same thing all over again, millions of times. Now that special groove where all the magic happens, that's called the active site. Its shape is no accident. It's perfectly complementary to its one specific substrate. This unique fit is the entire secret to an enzyme's power. And the key itself is, of course, the substrate. This is the molecule that's about to be changed. Maybe it's a big starch molecule getting broken down, or maybe it's two smaller molecules getting joined together. And this brings us to a super important idea, specificity. Because the lock and key are such a perfect, precise match, an enzyme that breaks down starch, like amylase, has absolutely no effect on a protein. It just can't. It's highly specific to its one and only job. And that's crucial for making sure the right reactions happen in the right places at the right time inside our bodies. Hey, we're about to dive into what happens when things go wrong with these perfectly shaped enzymes. But real quick, if you're finding this breakdown helpful for your revision, do us a favor and hit that like and subscribe button. It really helps us make more of these O-level explainers for you. All right, let's keep going. Okay, so we know how enzymes are supposed to work, but they are really, really sensitive. We need to understand what affects their performance, and first on the list is temperature. Just take a look at this graph. It tells the entire story of an enzyme's relationship with heat. You can see the reaction rate starts low, it climbs up to a peak, and then it just crashes dramatically. Let's walk through exactly why that happens. So when it's cold, all the molecules, the enzyme and the substrate, they have very little kinetic energy. They're moving slowly, they hardly ever bump into each other, so the reaction is super slow. Now, as we add heat, they gain energy, they start moving faster, they collide more often, and the reaction speeds up. But, and this is a big but, if it gets too hot, the enzyme itself starts to vibrate so violently that the bonds holding its perfect shape together actually break. The active site changes shape, and the lock is basically broken. So what's that sweet spot? What's that peak of the mountain on the graph? Well, for the enzymes in the human body, that peak performance, or optimum temperature, is right around 37 degrees Celsius. Yep, our normal body temperature. It's the temperature where they work the fastest and the most efficiently. All right, next up is another critical factor, pH. And just like with temperature, enzymes are incredibly picky about how acidic or how alkaline their surroundings are. And you'll see a very similar looking graph. Every enzyme has an optimum pH where it works best. But what's really interesting is that not all enzymes like the same conditions. Let's look at a really cool example of this. This shows it perfectly. Most of the enzymes in your body work best at a neutral pH, right around 7. But the protease enzyme in your stomach, it's a total exception. It actually thrives in the incredibly acidic environment of pH 2. It's perfectly adapted for its job in your stomach acid. So, what do we call it when the temperature gets too high or the pH gets too extreme? The enzyme undergoes denaturation. This is a word you absolutely have to know. It means the protein's special shape is broken, the active site gets permanently damaged, and the enzyme just can't function anymore. The lock is broken for good. This, by the way, is exactly why a really high fever is so dangerous. It can literally start to denature the critical enzymes that are running your body. Okay, I know that was a lot of information, so let's take a breath and just quickly recap the absolute must-know facts, the things you'll definitely need for your exams. Okay, so remember, enzymes are proteins that act as biological catalysts. Simple. Each one has a specific active site that fits a specific substrate, just like a lock and a key. They work best at their optimum temperature and pH, and if those conditions get too extreme, they will denature, and that change is irreversible. Those are the core rules of enzymes. So we've learned all about these incredible biological workers, the little machines that build and break down all the molecules in our bodies. But that kind of leaves us with one final, pretty fascinating question, doesn't it? If the enzymes are the workers out on the factory floor, what or where is the blueprint that tells the cell how to build each one so perfectly in the first place? Well, that's a question for our next explainer. Thanks so much for joining us. For more O-Level resources, check out Spotwork Learning. We're your O-Level success partner.